Oh, I didn't get the error with the speaker this time. All right, can somebody confirm you can hear me?
I can hear you.
Great, thank you. Let me share my screen. I don't think I've done that. Oh, it's still converting the lecture file, so this is going to be slow. All right, you should be seeing my screen now. All right, so today we're talking about Lab 9, identification of gram-negative organisms. Your infectious disease paper is due Saturday, August 17th, and that is this Saturday. So please finish up your infectious disease paper and turn it in by 11:59 p.m. Saturday, August 17th. If you have not cleared the project with me, you need to clear it with me, meaning you have to pick a project that nobody else has picked, meaning everyone's going to have a different infectious disease. And I also want to narrow it down in case you don't select something that's narrow enough. Like some people ask to do it on pneumonia, and I tell them, no, you can't do it on pneumonia in general, but you can do it on pneumonia caused by a specific agent, either a bacterial or a viral agent, like a species of bacteria. Any questions about the infectious disease project? All right. There was a question about the not Lab 9, identification of gram-negative organisms, but a question about the unknown bacteria. And let me show you the species. So it has to be one of the species in this table. And there's about 40 different species, about 20 gram-negative and 20 gram-positive. And I didn't count them exactly. Let's see, we're down to, yeah, there's 22 different species. Oh, there's only 22. So there's about 11 gram-positive, 11 gram-negative. Any questions about that?
Yeah, will that list be dated or that's the whole thing?
I don't understand your question.
Um, will there be more gram positive bacteria or it's only off of these 22?
It's only off of these 22. Okay.
Okay.
This was made in 2019, but we're using it today, if that's what your question was.
Yeah, I have some more questions, but it's regarding my number, so I'll just email you about it.
Okay. All right. Let's go on to Lab 9. I don't think I need this. I'm going to shut that down until my computer is working because it's only 74%. And let's go ahead and shut some of these down too, because it's having a hard time here. That can be shut down, I think. Yep. All right. Someplace I've got the lab here. Oh, that's the worksheet. All right, Lab 9, identification of gram-negative bacilli. This is the last lab for this course. You will need to complete your work and turn in Lab 9 worksheet. If you turn in the lab module, you may get a penalty. You'll also need to complete the worksheet and the case studies. There is some reading, not too much to today's lab, a little bit of one page in chapter 5 about biochemical tests and bacterial identification. And then you probably already read this, but chapter 10, methods of classifying, identifying microorganisms. page 272 to page 276. There are some video clips in today's lab. The learning objectives for today's lab: upon successful completion of this lab, you should be able to, 1, interpret the results for the following biochemical tests. The Imvik reaction. Imvik is actually 4 different tests. The indole test, the methyl red test, the Voges Proskure test, and the citrate test. I am not Russian, and I might be mispronouncing this test. I frequently just call it the VP test. Okay. We'll also talk about the carbohydrate fermentation test, the motility test, and the hydrogen sulfide production test. And then explain how to interpret the triple sugar iron test, the SEMS deep test, and then the test of growing your cells on McConkey's or MAC plates, and then hectone enteric agar or HE agar plates. Any question about any of those tests? And then 3, use biochemical analysis to solve case studies involving gram-negative bacterial infections. So this is looking at gram-negative species. Members of the family Enterobacteriaceae are all small gram-negative non-spore-forming bacilli. They're all facultative anaerobes. The Enterobacteriaceae is one of the largest families. There are 53 genera in this family. 24 of the genera have been known to cause human infections. Below are the 12 common genera causing clinical infections. All of the family of Enterobacteriaceae can ferment the sugar glucose, and they're all oxidase negative organisms. I'm sure you're familiar with a lot of them: Salmonella, Shigella, causing serious human disease. Escherichia, like E. coli, can cause disease. Citrobacter, Enterobacter, Serratia, Klebsiella, etc. Most of these genera are actually normal microbiota of the intestinal tract of animals, aka coliforms. Like E. coli is in our gut, Salmonella is in the gut of birds and some reptiles too. They are all opportunistic pathogens in that they can cause urinary tract infections, wound infections, pneumonia, and septicemia. So E. coli, when it's growing where it should be in your gut, it does not usually cause an infection. But if it's growing elsewhere in the human body, it will be an infection. Oh, good. My lecture Zoom page just finished, so my computer will work better now. Now, there are strains of E. coli which do cause infections even when it is in the gut. And we'll talk a little bit about those. So common genera causing opportunistic infections include Escherichia, like E. coli, Enterobacter, Klebsiella, Citrobacter, and Serratia. Like Klebsiella pneumoniae causes pneumonia when it grows in your lungs. Of course, anything that grows in your lungs will cause pneumonia. Several genera are associated with gastroenteritis, and these include the genera Salmonella, Shigella, and then E. coli Seravar 0157H7. If E. coli 0157H7 grows in your gut, it'll likely give you gastroenteritis. Any question about any of that? All right. This family Pseudomonaceae, such as Pseudomonas aeruginosa, is similar to the family Enterobacteriaceae, and that they are all gram-negative bacilli. However, the Pseudomonas, none of them can ferment the sugar glucose, and they're all oxidase positive. Remember, the Enterobacteriaceae family are all capable of fermenting glucose, and they're all oxidase negative. The Pseudomonas, especially Pseudomonas aeruginosa, can cause opportunistic infections, such as in especially immunosuppressed individuals, burn patients, and trauma patients. Pseudomonas aeruginosa does not usually get established in a normal individual, but in a burn patient or a trauma patient, it can bypass the skin barrier, or the wherever the trauma is, and cause an infection. So Pseudomonas aeruginosa is often implicated as a health-acquired infection or a nosocomial infection. It's important for a microbiologist to be able to identify and perform antibiotic susceptibility testing on gram-negative bacilli cultivated from a clinical specimen. In this lab, we're going to look at the biochemical tests that are used to identify gram-negative bacilli. And here's a bunch of gram-negative bacteria tests. We're not going to look at most of these, but we will look at some of them. The indole test is looking at the production of a bacteria that can produce the enzyme tryptophanase, converting the amino acid tryptophan to the products indole and pyruvic acid. What you do is you do overnight incubation, you add Covax reagent, which reacts with indole to produce a red color at the surface. E. coli tests positive for indole, and Klebsiella pneumoniae does not. So E. coli will be red and Klebsiella will not be in the indole test. This is what we're looking at, the amino acid tryptophan in the presence of tryptophanase can produce indole, which when you add Covax reagent, which stays at the top, it should be a reddish color. And this picture, for some reason, is not very red. It's more purple, but it should be a reddish color. All right. Any questions about the indole test? If not, let's talk about the methyl red and the Voges-Perskira test. I'll just call it the MR test and the VP test. These tests are usually started by inoculating one tube of the MR-VP media. And then we're going to look at the products of glycolysis. If the acid is produced from the glycolysis, we can add the methyl red, and if it turns red, it's positive because acid was produced in the methyl red test. On the other hand, if glycolysis produces a neutral product, you can determine that from the VP test, which will also turn red when the VP test is positive. So both methyl red and VP is looking at the metabolism of glucose in glycolysis. But MR is looking for the production of acid. VP is looking at the production of a neutral metabolite from glucose. As I stated, in the MR-VP broth, you normally have one tube, which is inoculated with your test organism. and then you grow that, incubate the tube, and then you split it in half. One half will be used for the MR test, one half will be used for the VP test. Methyl red test needs to incubate for 5 days. The VP test may be incubated only for 2 days, but usually if you're doing it together, you'll incubate it for 5 days. All right, and the VP test, I've already talked about the MR test. You add the dye methyl red, and if there's acid in the tube will remain red. If there is no acid in the tube will shortly become yellow. So you add methyl red, which is red, add it to the tube, it'll very quickly turn yellow. And that's methyl red negative, that's methyl red positive. In the VP tube, meaning the half of the tube for the VP test, you add Barrett's reagent A and Barrett's reagent B. You then shake it to add oxygen from the air, and then you let it incubate for about 60 minutes, looking for the production of acetone. Acetone is a neutral product, and if it is produced, the VP test will turn red, usually at the top. If the acetone is not produced, it will remain yellow. So VP and MR, if they're negative, they're both yellow, and if they're positive, they're both red. And so with the indole test, all three of them, if it's positive, it is red. Any question about any of that? All right. Let's move on to the citrate utilization test. This test is used to determine whether bacteria has the ability to use citrate as its sole carbon source, meaning in this tube, the only carbon source, as well as really the only energy source, is the sugar citrate. And if the bacteria can grow on the tube, you'll see growth on the tube, and the tube will turn blue or bluish. If the bacteria cannot grow on the tube cannot utilize citrate, and it will remain green. Remember, citrate is a sugar in the Krebs cycle. You don't need to know that in particular, but I'm just pointing out where citrate is in carbohydrate metabolism. All right, any questions about the citrate test? So it's blue and the test is positive, green if it's negative. You may not see growth in the green, and the blue you should see growth, although I don't see any there, but it should be blue. All right, I think we talked about the PR carbohydrate metabolism test. This is looking at the fermentation of one sugar. In the tube, you have one sugar, and it can be glucose or lactose or fructose or any other sugar. I've forgotten all the sugars we have in the table, mannitol and inulin, one other than lactose, as well as sucrose. If the tube sugar can be fermented, the tube will turn yellow. If the bacteria cannot ferment the sugar, the tube will either remain reddish or turn a pinkish color. If the sugar is fermented, it may be fermented without the production of acid, without the production of gas, sorry. In which case the tube, this is an inverted Durham tube, will be full of media. And if it does ferment the sugar, producing a gas, then you'll see a bubble in the inverted Durham tube. This we label negative, no fermentation. This we label A, meaning A for acid is being produced in fermentation, and this is labeled AG for acid and gas being produced. Now remember, in the carbohydrate metabolism test, you're looking at the metabolism, the fermentation of one sugar. And you can test all the different sugars. You just have glucose fermentation test, lactose fermentation test, etc. But it all falls under the carbohydrate metabolism test. And in the unknown project, you'll find it under carbohydrate metabolism. All right, let's move on to the motility test. In the motility test, we have a very low concentration of agar, and the bacteria can actually swim through the semi-solid media, meaning it isn't totally solid, it's semi-solid. And what you do is you stab inoculate the tube, and if the bacteria is not motile, it will only grow in the stab, as shown here on the left. If the bacteria is very motile, the bacteria will grow everywhere, and the tube will be cloudy everywhere. If the bacteria is only slightly motile, you'll see flaring away from the stab. So here we have the stab, and the bacteria will flare out from the stab. I'll make this one the same or similar, and then this will be a little different. Any question about that? So flaring of the bacteria, if it's slightly motile. If it's very motile, it'll just be cloudy everywhere. That's because the bacteria can easily swim through the semi-solid media. Remember, if bacteria are motile, they only have a flagellum or flagella. All right, let's move on to the hydrogen sulfide production test. Some bacteria can produce hydrogen sulfide gas. We can test for the production of hydrogen sulfide gas in at least 3 different tests: the TSI, triple sugar iron test, the sulfide indole motility, or the SEMS test, and the hectone enteric agar plate, or the hectone HE test. What happens is the production of hydrogen sulfide can react with a chemical added to the media, and it'll form a black precipitate. So if hydrogen sulfide is produced, you see blackening either in triple sugar iron, sulfide indole motility, or the HE plates. This looks like it's triple sugar iron and blackening. Usually in the tubes, it'll be the bottom of the tube, which will be turned black. And that's because the hydrogen sulfide will be produced anaerobically, not aerobically. And let's look at the HE plates. Another triple sugar iron where it's black. Here is the Sims tube where it's black, and the HE plates where there are black colonies because these colonies can produce hydrogen sulfide gas. Any question about that? Citrobacter, Salmonella, and Proteus produce hydrogen sulfide gas. All right, let's move on to the triple sugar iron test, also called the TSI test. This tube has glucose, lactose, and sucrose in it, and we'll look at the fermentation of glucose, lactose, and sucrose. But you'll notice that glucose is at a very low concentration, 1/10 the glucose compared to the amount of lactose and sucrose in the tube. And it has a phenol red indicator in the tube, as well as sodium thiosulfate, and ferrous sulfate, which can look for the production of hydrogen sulfide gas. With the TSI tube, you first get your needle and get the bacteria on the tip of your needle, and then you inoculate the butt of the needle, of the tube, meaning you stab the needle into the tube. You should actually go down a little bit lower than that. That's about halfway. You should go down about 2/3 of the way. You do not want to go down all the way, because if you go down all the way, you might tip the needle, touch the needle to the tube, in which case you can damage the needle. So you want to go down about 2/3 of the way. And what you're doing is you're inoculating the bacteria into the agar, and then this bacteria will grow anaerobically. And then you take the needle and pull it out, and then you get the bacteria on the tip of the needle again, which I'm trying to show you, and then you put the needle on the slant, and you make an S-like movement, inoculating the bacteria on the slant. This will look at growth of the bacteria aerobically. So in the TSI tube, we're looking at growth anaerobically and growth aerobically. Now, there's a number of things that can happen with the TSI tube. First, we'll be looking at the fermentation of any of the sugars. And if no sugar is fermented, the tube will remain red, both on the slant aerobically and in the butt anaerobically. If the tube can ferment lactose or sucrose, or one or the other, then the tube will be yellow on both the slant and the butt. If the slant is pink and the butt is yellow, this means that only the sugar glucose has been fermented, and glucose is at a low concentration in the media, and in the butt, the sugar is fermented slower, and so it has turned yellow. On the slant, the bacteria can grow rapidly and ferment the sugar rapidly, and all the sugar has been used up. Rather than die, the bacteria can utilize the protein in the media, and if you use protein, you will make ammonia from the protein, and ammonia is a base, and that will convert what used to be a yellow color for the fermentation of the glucose to a base ammonia, changing the color from yellow to red. If we were to look at this tube earlier, and you just do it overnight, so what would that be? About 18 hours. If you were to look at the tube like at 9 hours, you would probably see yellow on the slant because the sugar had been fermented, but all of the sugar in the slant got used up. And then the bacteria start utilizing the protein, making ammonia. The ammonia is released from the cell and it'll change the color of the media from yellow to pinkish or reddish. And that we call reversion. And this is what happens when only glucose is fermented. The butt will be yellow and the slant will be red. Any question about any of that? All right. And this tube here, it is black in the butt, and that's because hydrogen sulfide was produced. It is red on the slant, so this is only fermenting the sugar glucose, because if it was fermenting glucose or sucrose, it would be yellow on the slant. And you can't see it, but if it's black in the butt, that means it is fermenting the sugar, and only the glucose would be fermented because it's not fermenting sucrose or lactose. All right. And then there's one other thing you can look at, and that's the production of gas. If you see the breaking of the agar, a little bubble in the agar here, or a bubble in the bottom of the agar, then there's gas production in the fermentation. The interpretation of the TSI test is complex, and you just sort of have to learn it. Negative, black, hydrogen sulfide being produced, and you just know if hydrogen sulfide is being produced, there's at least one sugar that's being fermented. In this case, the slant is red, so only glucose is being fermented. This is what the tube would look like without hydrogen sulfide being produced and only glucose being fermented. And this is what the tube would look like if lactose or sucrose or both of the sugars are fermented as well as glucose. And let me just tell you, if lactose or sucrose are fermented, you know glucose is being fermented because glucose is the base sugar. But in this one, we don't know if it's lactose or sucrose or both of those sugars being fermented. And then the production of gas there. Any questions about the TSI test? All right, let's move on to the sulfide indole motility test. This test is looking at 3 different tests all in one tube. First, you add Kovacs reagent, and if the Kovacs reagent remains yellow or yellowish, then indole is not being produced. If, on the other hand, the Kovacs reagent turns pinkish or reddish, then indole was produced, and the tubes are indole positive. All 4 of these are indole positive. If the tube turns black, then hydrogen sulfide is produced. So hydrogen sulfide positive, hydrogen sulfide negative, all these others. And then the last test, SEM, is you're looking at motility. And you inoculate the tube. Let me blow this up a little. If the bacteria is only growing in the stab, then the bacteria is not motile. If the bacteria are growing outside of the stab, and this one is flared, especially right there, then the bacteria is modal, non-modal, non-modal. This one you can't see it, but because the black is everywhere, we can assume that this is modal. And then that's negative, negative control. And then SIM test stands for sulfur test, indole test, and then motility test. That's what the SIM stands for. Any question there? So 3 tests being run at once. All right, the MAC and HE plates are looking to see if the bacteria is gram-negative. If there's growth on a MAC or HE plate, the bacteria is likely to be gram-negative. About 95% of the time, it'll be gram-negative. And that's because the MAC plates and the HE plates have additives which select against gram-positive. You don't need to know the additives, but in the MAC plate, the additive is actually, oh gosh, what do you call that? Something your pancreas, bile salts. Bile salts and crystal violet. They get into the gram-positive cell and then prevent the gram-positive cell from growing. But gram-negatives are frequently going in the colon where bile salts are present, and they're not harmed by crystal violet because the outer membrane of the gram-negative cell prevents the crystal violet from getting in. So if there's growth, it's likely to be gram-negative. No growth on either MacConkey's or HE, it's likely to be gram-positive. Both HE and MacConkey's are also a differential plate, and they're looking at the production, fermentation of the sugar lactose. If the bacteria can ferment the sugar lactose on a MAC plate, the bacteria will be pinkish or purplish, and then the area around the plate will also be pinkish or purplish. If the bacteria cannot ferment the sugar lactose on a MAC plate, the bacteria will be their normal color, which in this case will be white or off-white. On the HE plate, if the bacteria can ferment the sugar lactose, the bacteria will be a salmon color. That's sort of a yellow-orange color. If the bacteria cannot ferment the sugar lactose, the bacteria will be their normal color, which in this case is white. And remember, on an HE plate, if the bacteria is black, either with fermentation or without, that means the bacteria is producing hydrogen sulfide gas. Any question about that? So HE and MAC plates are both selective for gram-negatives, meaning if there's growth, it's likely gram-negative. And then they're differential on the ability of the plate or the species to ferment the sugar lactose. All right, any question about Lab 9? While you're not physically going to be performing any of these tests, you will be held responsible for knowing how to perform and interpret these tests. We have some video clips for carbohydrate fermentation test, for the SEMS test, for the MR and the VP test. Remember, we normally inoculate that as one tube and then split it into two, one part used for the MR test and one part for the VP test. We have a little video discussing the TSI test and then a video talking about the MacConkey's auger. All right, let me take a look at this, see if it works. Um, Yeah. That link doesn't work anymore. So you're going to need to uh, look online or in earlier lab manuals to help fill in the step 1 to worksheet lab 9. So let's look at the worksheet. So you need to answer the following tests for worksheet 1. The oxidase test, the first cell has been filled in for you. So the positive result in the oxidase test, the cells or the spot will turn blue. So fill in the negative result. And then for the indole test, which is actually given in this lab module here, tell me what the positive result is and the negative result. And the MR test, tell me what the positive result is and the negative result. The MR, the VP, the citrate, these tests have all come from this lab module. So tell me what the positive result is and what the negative result is. And then in carbohydrate fermentation, tell me what the positive result will be if the sugar is fermented and what the negative result will be. Tell me what in the carbohydrate fermentation test, what the positive result will look like if gas is produced and what the negative result will be. And then moving on to triple sugar iron test, tell me what the two will look like if only glucose is fermented and then if glucose is not fermented. And then tell me what the two will look like if lactose and sucrose is fermented and the negative result if lactose and sucrose is fermented. I'm going to warn you here for the lactose glucose fermentation and the lactose sucrose fermentation, you need to state both the slant fermentation over the butt fermentation. Okay? If you only have one, you will only get partial credit. So tell me both the slant over the butt and here also the slant over the butt. The negative result should be the same, slant over the butt. Although you should know in the negative case, both the slant and the butt will be the same color in the negative case. And then tell me what color it will be if hydrogen sulfide is produced, positive result, and the negative result. And then if gas is produced, give me the positive result and the negative result. For the Sims agar, give me the positive result if sulfide is produced and the negative result. For the indole test, give me the positive result and the negative result. For the motility test, give me the positive result and the negative result. So actually all of these, the selective and differential agar from McConkey's and HE, tell me if the positive result if lactose is fermented and the negative result. You don't need the Microbiology website because everything we've been talking about comes from this lab module. Come on. So you don't really need to know microbiology because you can use this lab module to fill in table 1. Any question about any of that? All right, then complete the following. 2A, what reagent must be added to detect the presence of indole? If indole is present, meaning 2A1 and 2A2, if indole is present, the test will show what color? 2B, methyl red is the pH indicator that remains what color under acidic conditions? Methyl red pH indicator is used in what test? 2C, The wet test uses Barrett's A and Barrett's B reagents to detect the presence of acetone, a neutral product formed from glucose metabolism. If acetone is present, the test will show what color? 2D. Simmons citrate uses the pH indicator. So name the pH indicator. And that pH indicator turns blue under write one or underline or circle one, bold or acidic conditions. 2E, what is the pH indicator used to detect PR carbohydrate fermentation and is yellow under write one, acidic or basic conditions? You could also underline or highlight, I guess that's the word, or you can circle it. Okay. I'll give you a hint here. The name is what PR is an abbreviation for. All right. 2F. Bacteria that produce this structure test positive for motility. So what cell structure allows bacteria to be motile? 2G. Three organisms that produce hydrogen sulfide are species 1, species 2, species 3. And you could, if you don't want to do species, you could do a family, as long as the family is all positive. If an organism ferments glucose, but not lactose or sucrose, the TSI slant will be wet and the butt will be what color? State the three tests for which SIM is used. 1, 2, 3. MAC and HE agar plates are both what in that they inhibit the growth of bold or right gram-positive or gram-negative bacteria. And they're also what in that they separate various bacteria based on visual differences from sugar fermentation. Any questions about 2J? And then complete the case studies below using this key to solve for the gram-negative bacteria. So we have family Enterobacteriaceae here, and then family Pseudomonaceae here. And then you're going to be running different tests in your case studies. Fill in for case study 1 the information here. So if it gives the oxidase test, put the result there. If it ferments glucose, put the result there. Ferments lactose, put the result there. If TSI tube is used, that test is used for case study, put the results there. H2S production here. Motility test here. Indole here. MR test, VP test, and sodium citrate test. Now, some of these tests may not be used on the case study, in which case you just leave it blank. But fill in the tests on this table here, and then look up to determine which organism it is, and then write what species case study unknown number 1 is here, and then what species case study unknown number 2 is here, 3, 4, 5, there. I'll discuss case study unknown number 1, and then the rest I'll let you guys do. Just a minute, I'm trying to think where I've got it. Oh, maybe it's in the let's see if it's in the lab module. There it is, it's in the lab module. Case study 1. A prison inmate is brought into the ER room with an open wound. This prisoner is known for self-inflicting, meaning he damages himself. Routine culture and sensitivity test is ordered. The appropriate transport media is used for specimen collection and for inoculation of the prescribed media. Here we have the bacteria from the wound growing on a blood agar plate, and you can see the hemolysis pattern on that plate. Here we have the organism growing on a McConkey's plate, and it's growing on the McConkey's plate, so that should tell you something. And then the color tells you something as well. And then there's the Gram stain. You have to blow this up to look at it, because I can't tell what it is there, what shape it is. All right, it kind of looked like cockeye when it was smaller, but now that it is larger, I can see that's not cockeye. Just tell me if it's gram negative and what the shape of the bacteria are. And then some more tests were ran. The rapid test, it was catalase positive and oxidase negative. And then we performed some further tests on day 2. Maybe shrink that down a little. So A is the SIMS test with Kovacs reagent added. I don't see any bacteria, so motility we can't tell from this, but you can tell 2 of the 3 tests from that. And the MR test, the VP test, the SIM citrate test, and then a triple sugar iron test. Any question about any of that? Let me just briefly talk about case study 2. Mikey is a little boy and he talks his mother into buying a pet map turtle shown here. Mikey does very well at changing its water, but he's not necessarily diligent about washing his hands afterwards. So he'll handle the turtle and not wash his hands and then go and eat. About 2 weeks later, Mikey develops nausea, diarrhea, and fever. His pediatrician orders a fecal specimen and routine culture and sensitivity testing. Here we have his bacteria growing on a McConkey's plate. And because it's growing on McConkey's, it tells you something. And the colonies of interest are got an arrow added. And the color tells you something as well. And here we have a colony of interest growing on HE plate, the arrow looking at the colony of interest. And that tells you something because it's growing on HE and McConkey's, and then the color on both tell you something. The rapid test was done. The bacteria was catalase positive and oxidase negative. Some other tests were run. There we have the SIMS test there, the MR test, the VP test, the SIM citrate test here, and then TSI test here. So what species is causing Mikey's infection? Any question about that? Now remember, only write your answers in the worksheet to Lab 9. I don't even have it in the worksheet here. I mean, in the lab module there. All right, any questions about the case studies? So fill in this table, use this key to identify the bacteria, and only use this key for this lab. Don't use any other key. Do not use this key to identify your unknown bacteria. And then write your species causing infection here, and then submit the worksheet this Saturday at 11:59 p.m. And then hopefully I'll get it graded. By the way, I did get I think it was lab 5 graded. Let me stop the sharing. Then I can look to make certain it's lab 5. So lab 5, I think. Make sure I'm not sharing, because I'm sharing the grade book now. Yeah, I'm not sharing. Nope, it was lab 6 I got finished. So lab 6, the second grading will be due this Saturday. I'm going to try and work on lab 7 today. And if I get it completed tonight or very early in the morning tomorrow, then lab 7, the second grading will also be done this Saturday, meaning you have to get it turned in by lab 7. If you didn't get 100% on lab 6, you can turn in the lab for up to 100% by this Saturday. And I think lab 5 was last Saturday. Yeah, lab 5 has already been done. And so I get lab 5 graded for the second time sometime whenever I have time. It looks like most students didn't do lab 5, but okay. All right. Any questions about any of that? Any questions at all? If not, I'm going to turn it over to you. And if you have questions, please ask. I will be here to help you until 9 o'clock or the last student logs out. Lately, students have been logging out pretty early, in which case I will log out early as well. So if you have a question, make sure you log in before the last student logs out.